Good morning, everyone. Respected madams and sirs. Today we are here to have a seminar on the topic the stem cells, the research and therapy. Myself, Dr. Abir Lalshan, second year Physiology, PGT, Jorhat Medical College, and my moderator is Dr. Geeta Borua, Professor and HOD, Department of Physiology, Jorhat Medical College. So our learning objectives will be, first of all, we learn about the stem cells, the definition and sources. Next, about the stem cells in bone marrow. Then about hematopoiesis, the properties of stem cells, the types of stem cells, embryo cloning, which is also known as therapeutic cloning, some researches about stem cells. Next, we will know about the different stem cell therapies, and lastly, the summary and conclusion. So let's know about the stem cells definitions and the sources. The bone marrow contains a number of hematopoietic precursor cells and also acts as a storage pool of the mature cells before they are released into the peripheral circulation. All these blood cells that are derived from the pluripotent stem cells are also referred to as the hematopoietic stem cells. These hematopoietic stem cells, they produce the different blood lineages, which includes the red blood cells, the white blood cells, and also the platelets. The bone marrow is the richest source of the hematopoietic stem cells, whereas the peripheral blood also constitutes some of the hematopoietic stem cells, and from the peripheral blood they are mobilized from the bone marrow to the peripheral blood by administration of granulocyte colony stimulating factor or granulocyte monocyte colony stimulating factor. The umbilical cord blood collected following delivery is also a rich source of hematopoietic stem cells. So this is the diagram which we discussed in the last slide. That is the red bone marrow, which acts as the storage for the different hematopoietic stem cells, from which the different blood cells of different lineages such as the red blood cells, white blood cells, and the platelets are produced. So these are the general steps of hematopoiesis. First of all, from the stem cells they gets converted to the progenitor cells, and from there to the intermediary precursor cells, and from there the mature cells are formed. So continuing with the process of hematopoiesis, the mother hematopoietic stem cells is the pluripotent stem cells, which is also known as hemocytoblast. These pluripotent stem cells is multilineage stem cells that are capable of producing two types of stem cells, the myeloid and the lymphoid stem cells. The lymphoid stem cells are unipotent stem cells that produce only lymphocytes, whereas the myeloid stem cells are again pluripotent in nature that produce the red blood cells, the granulocytes, the monocytes, and the platelets. Thus, in the bone marrow, there are two types of cells, the stem cells and the progenitor cells. From the stem cells, they develop into the progenitor cells. So this is the diagrammatic view of the different types and the stages of hematopoiesis and the major cytokines influencing them. So as we discussed in the last slide, from the pluripotent stem cell they give rise to the myeloid stem cells and the lymphoid stem cells. As we said, that lymphoid stem cells being the unipotent stem cells, they give rise to only B and the T lymphocytes, whereas the myeloid stem cells being the pluripotent stem cells, they, in the presence of different interleukins and other stimulating factors and cytokines, they produce step by step, first of all the multipotent progenitors, then they get converted to the committed, that is the unipotent progenitors. From there they give rise to the precursors in the form of pronormoblast, then the myeloblast, the monoblast, and the megakaryoblast, and lastly they give rise to the mature forms, which are respectively the erythrocytes, the eosinophil, basophil, neutrophil, monocyte, platelets from the myeloid stem cells. In this way the stages of hematopoiesis is completed. So let's know about the properties of stem cells. There are two important properties of stem cells in the bone marrow. This includes the self-renewal and the differentiation. So by the self-renewal property, the stem cells are capable of duplicating themselves. By this property, they physiologically never die, and their stock or their storage in the bone marrow remains abundant. So the bone marrow never goes out of stock for the stem cells. Whereas by the property of differentiation, they develop into specific lineage of cells. That is, the stem cells gets differentiated into progenitor cells of various cell lines, that give rise to specific blood cells. So this slide is about the scientist contribution led by Dr. John Edgar Dick, who was the first to study the details of properties and the nature of the stem cells. He transformed the study of human hematopoiesis and leukogenesis, which is development of the different methods for transplanting human bone marrow into immunodeficient mice, resulting in multilineage repopulation of the murine bone marrow and other hematopoietic tissues. By this approach, he identified long-term repopulating human hematopoietic stem cells and generated mouse models of leukemia. So this is the slide about the different types of the stem cells that we already discussed. The two classifications of stem cells are mainly the myeloid and the lymphoid stem cells. The myeloid stem cells, as we said, are pluripotent, that is the trilineage in nature, that give rise to different progenitor cells. This includes the cells of the erythroid series, the cells of the granulocyte progenitors, that includes granulocytes in the form of neutrophils, eosinophils and basophils, and the monocytic progenitors from the monocytes, whereas the megakaryocytic progenitors that forms the platelets. From the lymphoid stem cells, which are unipotent in nature, they give rise to the different cells of the lymphocyte series. So this is the diagrammatic view of the development of leukocytes. From the pluripotent stem cells, first we see they give rise to the myeloid stem cells and the lymphoid stem cells. As the myeloid stem cell is trilineage in nature, they give rise to the three forms of blood cells, which includes the first of all the granulocyte monocyte progenitor, which leads to formation of neutrophil, basophil, eosinophil and monocyte, and from erythroid progenitor that leads to red blood cells, whereas from the megakaryocyte progenitor that gives rise to the formation of platelets. Whereas if we consider the lymphoid stem cells, they give rise to prolymphocyte as a precursor, and from there they are differentiated into B and the T lymphocytes and the natural killer cells. So this is the complete picture of the steps of leukopoiesis. So as we see, the pluripotent stem cells give rise to the myeloid stem cells and the lymphoid stem cells, while the lymphoid stem cells being unipotent, they give rise to only lymphocytes in the form of T and the B lymphocytes, and pre-T cells and the pre-B cells being the precursors. Whereas if we consider the left side of the diagram, which includes the myeloid stem cells, they gets converted to CFU-GE-MM, that is the colony forming unit, the granulocyte, erythroid, megakaryocyte and the monocyte, and they respectively give rise to CFU-E, that is converted to erythroid series, CFU-mega, which gets converted to the platelet series, giving rise to the platelets, whereas the CFU-GM, that is the granulocyte monocytes, they get separated, and from this ultimately we get the different granulocytes in the form of neutrophil, eosinophil and basophil, and the CFU-M, which gives rise to monocytes in the form of macrophage and dendritic cells. Thus it completes the procedure of leukopoiesis. So this slide is about the details of the development of T cells and B cells. As we see, from the common lymphoid progenitor, they give rise to pro-B cell and pro-T cell, also the NK cell progenitor. From the left side of the diagram, we see that pro-B cell, they step by step gets converted to pre-B cell, then to naive B cell. The naive B cell, in one part it's continues to form mantle cell, the centroblast, the centrocyte, which give rise to memory cell, marginal zone B cell and the IgA and IgE plasma cell. Whereas on the other side, the naive B cell, they gets converted to B immunoblast and IgM plus plasma cell. If you consider the pro-T or the NK cells, they get similar steps. They gets converted to first of all the pre-T cell, then to naive T cell, and from there is two classification, one into the CD8 plus T immunoblast and the other into CD4 plus T immunoblast, which respectively gives rise to the CD8 plus T cell and the CD4 plus T cells, whereas the NK cells is also produced from the NK cell progenitor. This is the complete picture of the development of T and the B cell and the NK cells. In this slide, we will know about the stem cells related immunodeficiency states. there are mainly two types of immunodeficiency states related to stem cells. These are the autosomal recessive severe combined immunodeficiency, and the other is the X-linked severe combined immunodeficiency. In the first type, that is the autosomal recessive severe combined immunodeficiency, there is actually the stem cells, that is mainly the pre-T cells in the thymus, they fail to develop into the mature T cells, and as a result of which there occurs severe deficiency in cellular immunity. Whereas in the X-linked severe combined immunodeficiency state, the pluripotent stem cells fail to develop into the progenitor cells of the lymphoid series. Therefore, as the whole lymphoid series suffers, we get deficiency in both the development of humoral and cellular immunity. So before going to the stem cells research and therapy, we will lastly know about the different stages of thrombopoiesis. So to start with, the pluripotent stem cells, which gets converted to the committed stem cells, from there to the CFU mega, and from the CFU mega to the megakaryoblast to the promegakaryocyte, which is also known as the basophil megakaryocyte. Next forms the granular megakaryocyte, which leads to the formation of mature megakaryocytes, and lastly the platelets. In this way, the thrombopoiesis steps are completed. So let's discuss about the bone marrow transplantation. The bone marrow transplantation, also known as BMT, is the process of collection and infusion of hematopoietic stem cells obtained from the bone marrow or peripheral blood of either of the same individual hematopoietic stem cells or from other individuals. The scientist who was mainly involved in this bone marrow transplantation was Dr. E. Donald Thomas, who received the Nobel Prize in Physiology and Medicine in 1990 for bone marrow transplantation. He and his colleagues pioneered the successful use of bone marrow transplantation. In this procedure, there is replacement of the cancerous cells and the stem cells that are damaged by chemotherapy and radiation by the donated healthy cells, which engraft within a patient's bone marrow. The discovery was a cure for leukemia and other blood cancers and earned Dr. Thomas the 1990 Nobel Prize in Physiology or Medicine. So in this slide we will learn about the embryo cloning, also known as therapeutic cloning. The therapeutic cloning refers to the production of human embryos for use in research. This process is aimed at producing stem cells which can be used to study the human development and to treat disease rather than to create a cloned human being. The stem cells that are extracted from the egg at the blastocyst stage of development can be used to generate any type of specialized cells in the human body. The first human embryo for the purpose of therapeutic research was cloned in November 2001 by the scientists from the Advanced Cell Technologies in Massachusetts. The application of it includes the stem cells can be used to serve as replacement cells to treat heart disease, Alzheimer's disease, cancers, etc. So this is the diagrammatic view of the embryonic stem cells production from the blastocyst stage. The blastocyst, which is obtained after fertilization of the oocyte with the somatic cell, and from there the embryonic stem cells which are produced is used for different disease treatment like the heart disease, the Alzheimer's disease and cancers, but the red line in the bottom shows that the embryonic stem cells produced in these methods are not used for producing a cloned human baby. So one of the new strategies for greater potential for research and treatment using the stem cells include the EPSC, which is also known as the expanded potential stem cells. Unlike the embryonic stem cells, the expanded potential stem cells are produced from the 8-cell embryo and which then can be used for use in placenta, yolk sac and all cells of the body. Whereas, as we discussed, the embryonic stem cells are produced from the blastocyst stage and can also be used for all the cells in the body. And another process, there is the induced pluripotent stem cells, which also known as IPS, is produced by genetic reprogramming by inducing the transcription factors, and then that can also serve for all the cells in the body. Here we will learn about the bone stem cells. It is seen that whenever there is a fracture of a bone, then it leads to maximal activation of the periosteal and the intraosseous osteoblasts, which are involved in the break. So these new osteoblasts are formed almost immediately from the osteoprogenitor cells, which are the bone stem cells that lines the bone, also known as the bone membrane. Therefore, within a short time, a large number of osteoblastic tissues and new organic bone matrix are formed between the two broken ends of the bone, which is known as the callus. Many orthopedic surgeons use this phenomenon of bone stress to accelerate the rate of fracture healing by accelerating the osteoblastic activity at the break, which often shortens the convalescence. So this diagram shows how the fracture of a bone leads to the formation of the bone stem cells that lines the bone, also known as the bone membrane, and leads to increased osteoblastic activity. This increased osteoblastic activity leads to the callus formation, and then by the process of bone remodeling helps in the formation of the new bone. So this slide is about the induced pluripotent stem cells, also known as the iPS cells. Here the 4 transcription factors are reprogrammed into the cells to produce the induced pluripotent stem cells. These iPS cells could be used to generate many cell lines for different disease, and so that the mechanism of the disease can be studied. These are also used to test how different drugs work on those diseases, to test the effectiveness and toxicity of drugs on the cells from people with genetic differences, and also to produce stem cells that can be used to treat many diseases. The iPS cells can treat specific disease including the sickle cell anemia and Parkinson's disease, which has been successful in mice, and an attempt to treat the macular degeneration with these iPS cells in humans will soon be made. So in this diagram we see the procedure of somatic cell nuclear transfer, as we said already, how it is produced from the blastocysts, and in the second diagram we see the iPS cells production, that is done by inducing the transcription factors, and by the process of direct reprogramming the iPS cells are produced. So this is another diagram showing how the iPS cells are produced from the adult cell with the help of the iPS reprogramming factors, and once the iPS cells are produced, they can be used for different cell lineages across the mesoderm, endoderm and ectoderm, respectively. The mesoderm including the cardiac muscle cells, the skeletal muscle cells, the tubule cells of the kidney, red blood cells, and the smooth muscles of the GIT, whereas the endoderm internal layer including the lung cells, the thyroid cells, and the pancreatic cells, whereas the ectoderm including the skin cells of the epidermis, neuron cell, and the pigment cells. So let's move on to some researches about stem cells. To start with, the microglia that are derived from the erythromyeloid stem cells migrate into the CNS early in development. They play an important role in immune responses within the central nervous system. In addition to the role of the stem cells in immune response, recent evidence suggests they are also active in healthy brain tissue development and may also have important roles in normal brain development and function, including the pruning, that is the removal of the excess synapses which are formed during development, and synaptic plasticity. So we will be discussing now about the knockout mice. The knockout mice is the experimental mice which is produced by the p53 gene, which is also the tumor suppressor gene, knocked out. Results showed that when the p53 gene is knocked out, all the experimental mice develop tumors. The 2007 Nobel Prize in Physiology or Medicine was therefore awarded to the scientists who developed knockout mice to find out the role of p53, which is the tumor suppressor gene. This is done actually by using the mouse embryonic stem cells grown in vitro. First of all, a defective copy of the gene is made and introduced into the embryonic stem cells, which are then put into a normal embryo. The mouse that develops is a chimera, a mixture of the normal and the mutant types. Because this chimerous tissue contains the cells with the inactivated gene, that is the p53 knocked out, this mutation is also present in its gametes, that is the sperm or the ova. So when this mouse is mated with a wild, that is the normal type mouse, some of the progeny will have the targeted gene, that is the p53 gene, knocked out. So this has been widely used to determine the physiological importance of many gene products, including the p53 gene. So this diagram is showing the knockout mice. Here we see the mouse embryonic stem cells are genetically engineered to disrupt the gene, and once the blastocyst is produced, it is transferred into the uterus of host female. So the resultant mouse contains a mixture of genetically engineered cells and inner cell mass cells. So this is the basis of knockout mice. So moving on to the germinal stem cells research. The stem cells that can divide indefinitely include the germinal stem cells that gives rise to ova and the sperm, and the hematopoietic stem cells present in the bone marrow. They have an enzyme called the telomerase which duplicates the telomere DNA. Most of the cancer cells we know also produce the telomerase enzyme. So this is responsible for the ability of the cancer cells to divide indefinitely. This telomerase enzyme consists of an RNA portion that contains nucleotide bases complementary to the telomere DNA, and a protein portion that acts as a reverse transcriptase enzyme producing the telomere DNA using the RNA as a template. So because of this significance of telomeres and telomerase enzyme in the physiology, cancer and aging, the 2009 Nobel Prize in Physiology or Medicine was awarded to 3 scientists who were instrumental in the discovery and germinal stem cells research. Some researches have also been done for the stem cells used in myocardial infarction. There are some stem cells found in the heart which normally replace about 1% of the myocardial cells per year, but they are not sufficient to repair a myocardial infarction. So they have been multiplied in vitro and introduced into the heart patients with some success. So part of the 2012 Nobel Prize in Physiology or Medicine was awarded to Shinya Yamanaka for pioneering work first published in 2006. He and his colleagues demonstrated that the adult human fibroblasts could be changed into pluripotent stem cells by using retroviruses to insert them into the cell's DNA with 4 human genes coding for transcription factors. So now we will be discussing about the different therapy in which the stem cells are used. First of all, in Parkinson's disease. So before the dopaminergic neurons are completely lost, the administration of levodopa can relieve some of the motor deficits in Parkinson's disease. Levodopa is a precursor of dopamine and can cross the blood-brain barrier. So scientists produced the iPS cells from the patients with Parkinson's disease, removed the 4 transducing genes from the iPS cells, and then induced them to develop into dopaminergic neurons. Such neurons can be used to know about the sporadic form of Parkinson's disease, which is the most commonest form and may be hopefully used for the basis for future treatments. Secondly, the use of stem cells at the site of brain injury. The plastic changes can occur after injury to the brain in adults. Sprouting of new axons can help to repair the damaged CNS. However, the sprouts do not necessarily restore the normal functioning of the CNS. Therefore, research is being carried out to explore the potential of human embryonic stem cells for restoring the CNS function. Administration of neurotropins shows some promise in experimental animals, and so the implantation of embryonic stem cells can be useful at the site of injury. Next comes the use of the stem cells in spinal cord injury. It has been seen that certain proteins called NOGO, which is produced by oligodendrocytes, they inhibit the axon regeneration in the CNS. This inhibition was demonstrated by a improved axon regeneration in rodent models of spinal cord injury when there were some antibodies that neutralized the NOGO action was used. So despite the barriers to the axon regeneration in the CNS, scientists recently came out with the idea that the transplanted embryonic neural stem cells, when they are suspended in a cocktail of growth factors, could sprout thousands of axons to restore the hind limb function in rats with transected spinal cords. These studies of potential stem cell therapies for human spinal cord injuries offer some hope, but should be interpreted cautiously. In this slide, we will be discussing about the stem cell therapy in myocardial infarction. In recent years, scientists have come out with a number of potential stem cell therapies for myocardial infarction. These include the use of stem cells from the bone marrow that promote healing, the possible differentiation of embryonic stem cells and iPSCs into myocardial cells, and also the transformation of fibroblasts into myocardial cells. Another approach has also been done to stimulate the myocardial cell division, which is normally too limited to repair the infarct, which has been done in rodent hearts, but More research is required in these strategies to include in human hearts before they become medical therapies. So this diagram is about the new strategies involved regarding the stem cells in the treatment of myocardial infarction. Some of them include the use of embryonic stem cells, induced pluripotent stem cells, skeletal muscle stem cells, bone marrow cells, mesenchymal stem cells, cardiac stem cells, and the stem cell derived exosomes. Some of them have already been discussed in the last slide. So this slide is regarding the stem cell therapy in wound repair. Some recent studies conducted regarding tissue regeneration in certain organisms with simpler development showed that there is some activity of the mesenchymal stem cells during the process of wound healing. These blastemal cells are positioned to interpret the cues from the environment and to execute decisions about the direction of wound repair. Whereas in mammalian wounds, the stromal stem cells appears to be positioned to perform the functions similar to those of the blastemal cells. This includes the communication with both the overlying epithelium and the inflammatory cells in the mesenchyma. So the stem cells are now also being used for motor neuron diseases. It is found that the induced pluripotent stem cells derived from the fibroblasts of a person with amyotrophic lateral sclerosis can differentiate into motor neurons. So these reports and others encourage the hope for the potential therapeutic benefit of the induced pluripotent stem cells in regenerative medicine. So the stem cell therapies are now also being tried for the treatment of diabetes mellitus. Besides the traditional treatment with insulin and other oral hypoglycemic drugs, some clinicians also try to improve patients' care by using stem cell therapies, using the embryonic stem cells, the induced pluripotent stem cells, and the mesenchymal stem cells. So to start with the embryonic stem cells, they display a virtually unlimited plasticity, including the differentiation into insulin-producing beta cells. But they raise ethical concerns just like induced pluripotent stem cells due to the risk of tumors. The induced pluripotent stem cells may further inherit somatic mutations, but there is incomplete reprogramming which allows the generation of patient or the disease-specific cell lines. The mesenchymal stem cells avoid such issues but not successfully differentiated into beta cells. So instead of these, the mesenchymal stem cells and their pericyte phenotypes have been recognized to secrete numerous immunomodulatory and other tissue regenerative substances. This diagram shows the use of stem cell therapies in diabetes mellitus treatment. So here we see the induced pluripotent stem cells that helps in the regeneration of beta cells, and also the MSCs, that is the mesenchymal stem cells, which produce some immunomodulating substances that lowers the insulin resistance, that lowers the immune rejection, and also lowers the low-grade inflammation. So let's discuss about the mesenchymal stem cell therapy in cancer. A meta-analysis has found that 1012 participants who received mesenchymal stem cell for the treatment of ischemic stroke, Crohn's disease, cardiomyopathy, myocardial infarction, graft versus host disease has proved to be healthy volunteers without any indication of malignancy. Meanwhile, research has employed the recruitment of mesenchymal stem cells to tumors in order to target the malignant disease with genetically modified mesenchymal stem cell. For example, by overexpressing pigment epithelium derived factor to reduce the angiogenesis, or by overexpressing the tumor necrosis factor related apoptosis to induce the apoptosis. So despite using the mesenchymal stem cells from different sources, the data has found that there is reduction in tumor growth, and therefore prolongation in survival of the rodents. Due to this success, they initiated the first in-man trial, TREATME-1, which is aimed to target against advanced gastrointestinal cancer. So this slide shows the use of mesenchymal stem cells in cancer therapy. Here we find that the mesenchymal stem cells induces the immune function through the T cell lymphocytes, which inhibits the angiogenesis, and as described in the last slide, it is useful in the treatment of different types of cancer. So in this slide we will discuss about the regenerative medicine, which involves developing future medical treatments using stem cells. The embryonic stem cells, which is derived using a patient's nucleus, is inserted into an unfertilized oocyte's cytoplasm, which can develop into tissues tolerated by the host's immune system. Similarly, the induced stem cells, which is derived from a patient's fibroblasts, it produce differentiated tissues that won't be immunologically rejected. So there are some differences between iPS cells and the embryonic stem cells, including a large number of sites where the 4 transducing factor genes are inserted by the virus into the host DNA in case of the iPS cells. So this might cause genetic disruptions and can induce tumor formation and cancer in case of the iPS cells. So this diagram shows the use of regenerative medicine. In the first we see the embryonic stem cells, which are produced from the embryo, which after differentiation is used for multiple cell types. Then comes the hematopoietic stem cells, which are developed from the bone marrow, but they are used for blood cells only. And lastly we see the induced pluripotent stem cells, which after reprogramming and differentiation is used for multiple cell types. In this way, the regenerative medicine has brought a renaissance in the treatment using the stem cell therapy. So this slide we will discuss about the cord blood banking. The blood from the umbilical cord contains stem cells which can be used for later purpose in life. For example, if a child is having leukemia, receiving chemotherapy, then the child might need them in future to reconstitute a hematopoietic system if the bone marrow has been destroyed by chemotherapy. So this cord blood banking is a risk-free procedure, but it is an expensive one with a low probability that it will someday be used. So this picture shows the procedure of cord blood banking. In the first step, first of all the baby is born with the umbilical cord and the placenta attached, and then the cord is clamped and cut. As we know, the umbilical cord is a rich source of the stem cells. The blood drawn from the clamped cord is collected in a special collection bag. Then it is quickly delivered into the laboratory for examination and analysis of the blood, and then the blood is stored in the special container, which is kept in liquid nitrogen storage tank inside a secured facility for future purpose. So going through the last few slides of our discussion, we'll discuss here about the embryonic stem cells production. The scientists recently reported success producing the embryonic stem cells from an enucleated human oocyte and a diploid nucleus from a donor fetal or baby skin cell. However, there is low probability of producing healthy children by this technique. The part of 2012 Nobel Prize in Physiology and Medicine was awarded to John Gordon, who first demonstrated that a somatic cell nucleus from a frog intestinal cell transplanted into an enucleated egg cell could develop into a normal adult frog. So continuing with the embryonic stem cells, the success of somatic cell nuclear transfer demonstrated that the genes are not lost during the development, and the effects of aging experienced by the somatic cell nucleus must be reversed in the egg cell cytoplasm. So the latter effect is also true for the normal fertilization, and the cellular age of sperm and eggs, which is the same as the other cells in the parents' bodies, is reset when the fertilization occurs, so that the embryo's cellular aging clock starts over. So this slide is the recapitulation of how the embryonic stem cells are produced from the blastocyst stage, and then their classification across the different cell line in the form of ectoderm, mesoderm, endoderm, and germ line. From the ectoderm, they forms the brain and the skin cell layer. From the mesoderm, they form the muscle, the blood, the bone, and the cartilage. Whereas the endoderm forms the lung, the gut, and the liver. And from the germ line, they forms the sperm and the egg. So this constitutes the embryonic stem cell form across the different germ cell layer. So coming to the summary of our discussion, we came to know that there are two ways that the stem cells can be produced. One is by somatic cell nuclear transfer, that is the SCNT, taking the nucleus from a differentiated cell, that is such as the fibroblast of a patient, and then transferring it to a cytoplasm of an enucleated egg. The newly formed diploid egg cell develops into blastocyst, from which the pluripotent embryonic stem cells are derived. The other technique is using a cocktail of transcription factors and then producing the iPS cell. However, scientists have found some subtle differences in the degree of DNA methylation between these two different types of patient-derived pluripotent stem cells and between each of those and the pluripotent blastocyst cells formed following normal fertilization. So to conclude, more research is required before the promise of regenerative medicine using these technologies. In an effort to circumvent the problems with the pluripotent stem cells, scientists have tried to convert one differentiated adult cell directly into another, a process known as transdifferentiation. The different groups have reported different degrees of success at converting the fibroblasts into dopaminergic neurons, glutamine-releasing myocardial cells and others. However, as with the two methods of producing the patient-specific pluripotent stem cells, more research is required using this transdifferentiation process, which can be safely used in regenerative medicine in future. So with this, we come to an end of our discussion. Thank you all for your patient hearing. Thanks a lot.